Hi everyone, I'm Ayah Khalid and I will be talking about T-cell mediated immunity and lymphocyte development. So that we can understand lymphocyte development, we must understand the lymphoid organs, in which we have primary lymphoid organs and secondary lymphoid organs. As for the primary lymphoid organs, we have the bone marrow and the thymus, in which we have the development and maturation of lymphocytes. Although they do mature in these lymphoid organs, we still consider them naive since they have not encountered any antigens yet. As for the secondary lymphoid organs, this is what allows the interaction of lymphocytes and antigens, and this is beneficial since we have antigen-presenting cells patrolling around the body, and they collect antigens from all over the body and move on to the lymph nodes and present these antigens to lymphocytes, allowing them to become activated and perform their effector functions. And as we can see here, these few lymphocytes, they must be able to locate microbes that enter and reside anywhere within the body, and this is through the action of antigen-presenting cells such as dendritic cells, B cells, and macrophages. Most importantly, we have dendritic cells, which, as we know, are the bridge between the innate immune system and the adaptive immune system. So our main focus is going to be about T lymphocytes, which are a major component of the adaptive immune system, and they play an important role in cell-mediated immunity. So how do we differentiate T lymphocytes from other cells? It's through the presence of specific membrane-bound marker proteins or cell surface proteins known as cluster of differentiations. So these, we have several of them. We have CD4, CD8, and CD28. So CD4 and CD8 are co-receptors which are going to stabilize the binding of TCR with MHC proteins, which is one of the stimulatory signals for T cell activation. Whereas CD28 is also involved in the activation of T cells. So this is sort of a representation of what a T cell looks like. So it will always have a TCR and a CD28 on its surface. And over here, this one has a CD4, but this may also be a CD8. And yeah, these are the cluster of differentiations that we will always find within the T cells. And for CD4 cells, we have different subpopulations which are dependent on the cluster of differentiation proteins that they express and the cytokines that they release. And these include T helper 1, T helper 2, T helper 17, and regulatory T cells. Regulatory T cells we're not going to pay much attention to within this lecture. Okay, so as we mentioned, T cell receptors, it plays a role in the activation of the T cell, and this is what is going to enable the T cell to bind to its specific antigen or a specific epitope. And it is actually a complex of proteins rather than just one protein, and the specific antigen or epitope must be displayed on the surface of an antigen-presenting cell so that it can be recognized by the T cell receptor. So the antigen fragment has to bind to the major histocompatibility complex molecule, or MHC, on the surface of the antigen-presenting cell in order to be recognized by the TCR. And we're going to see more details about this in the coming slide. So over here, we can see the interaction between the antigen-presenting cell and the T cell through its MHC and TCR respectively. So as for MHC, we have two classes, as we can see over here. We have MHC1 and MHC2. MHC1 is responsible for displaying or presenting endogenous antigens, so antigens that are coming from within the body, whereas MHC class 2 is responsible for presenting exogenous antigens. In both cases, we can see that the MHC has a cleft in which it holds the antigenic peptide, and this is going to allow its recognition by your TCR. And earlier, we mentioned that CD4 and CD8 are co-receptors that are going to be helping with the binding of MHC to TCR. So in this case, CD4 and CD8, they're going to stabilize the interaction and they're going to enhance signal transduction. And we can always think of it like this. We have MHC2 is always going to be related to CD4. So 2 times 4 is 8. And MHC class 1 is going to be related to CD8. And we can also think of it as 1 times 8 is also 8. And earlier we mentioned that CD28 also plays a role in the activation of the T cells. But this is only our first signal, and we require 2 signals for the activation of T lymphocytes, as we're going to see right now. So what we have on the surface of the dendritic cell, we have an endocytic PRR, which is a pattern recognition receptor, and we have toll-like receptors. So let's take it one by one. So our endocytic PRR is going to bind to the pathogen, and as the name suggests, it's going to endocytose or engulf the pathogen. So after engulfing the pathogen, it's going to present it on the cleft on its MHC protein, and this is going to allow recognition by the TCR, and this gives us our first signal. The second signal is due to our toll-like receptor and PAMP, or pathogen, and PAMP is the pathogen-associated molecular pattern, or the pathogen binding to the toll-like receptor. This binding is going to stimulate the upregulation of B7, which is also known as CD80 or CD80/86, and this is going to bind to CD28 on the surface of the T cell. Now, the presence of both of these signals together is going to stimulate the differentiation and proliferation of the T cell. And as we can see here, it's differentiating to either a T helper 1 or a T helper 2 cell. And this slide sort of summarizes what I've just explained. So we have the endocytic pathway, which enables the dendritic cells to capture, process, and present antigens to T cells on their MHC2 proteins. Whereas the TLR signaling, it provides the activation signal to DCs, which triggers co-stimulation, which is our second signal, which is our co-stimulatory signal. And we said that we require both antigen recognition signal, which is signal 1, and co-stimulation, which is signal 2, to fully activate and differentiate the T cell. Now, in the case that we have MHC binding to TCR, which is our first signal, but we do not have B7 and CD28 binding, which is our co-stimulatory signal, there would actually be no T cell response. So there would be no activation, no proliferation, and no differentiation. So antigen presentation without the co-stimulatory signal will lead to anergy, which is cell inactivation. So now what happens after we get these two signals and the T cells are activated? We're going to have proliferation and differentiation. So proliferation is the expansion of the number of the antigen-specific cells, whereas differentiation is going to convert them into effective defenders. So what happens is the T cell itself is going to secrete cytokines such as interleukin-2. And this interleukin-2 is going to bind to the IL-2 receptor on that same cell. So after it secretes it, it's going to bind. So it'll look something like this. So it secretes it and it binds onto the surface of the receptor over here. This is going to stimulate the proliferation of the antigen-specific cell, which is going to give us multiple clones of that T cell that are specific to the antigen that we're trying to fight. Then they may differentiate into either effector T cells or memory T cells. So effector T cells are going to be responsible for fighting the current infection, whereas the memory T cells are going to reside within the body in case of any reinfection. They are available to fight immediately and the response would be faster. So effector cells, we have effector functions that are different to each CD4 or CD8 cell. So it can be activation of macrophages, B cells, or other cells in the case of CD4 cells, since they are mainly CD4 helper cells, whereas CD8 cells are going to kill infected target cells and they are going to activate macrophages since they are more cytotoxic and they act directly and kill directly. And again, this sort of summarizes what I have just explained. So the T cells are going to respond by secreting interleukin-2 and expressing the receptors for interleukin-2, which leads to an autocrine pathway of cell proliferation. An autocrine is basically when the cell acts on itself. So it secretes something that was going to act on the same cell. And then this is going to result in the expansion of the clone of the T cell that are specific for the antigen, and some of them are going to differentiate into effector cells, whereas some of them are going to differentiate into memory cells, which survive for long periods and are present in the case of any reinfection with the same antigen. So the first encounter of a naive T cell with an antigen, which converts it into an activated T cell, is known as T cell priming, and this includes all the steps that we mentioned earlier. And as we said, after a T cell is activated, it is going to start to secrete different cytokines depending on its subtype or subclassification as a T cell. So cytokine signaling is the method of action of the cytokines. So as we said, they have different functions, so their cytokines act in different ways and act on different cells. So as for cytokine signaling, we have three mechanisms. We have autocrine, paracrine, and endocrine. As for autocrine, it's what we discussed about interleukin 2. It is when the cell acts on itself. So this is what happens when we have the secretion of interleukin 2 and then binding of that interleukin 2 to the receptor on the same cell. Now paracrine is when the cell acts on a cell within its proximity, so it's a nearby cell within the same tissue. So as for interleukin 2, although we said it is mainly autocrine, it may also act as paracrine, but its major effect is autocrine, so it mainly acts on the same cell. As for endocrine, the cell is going to act on a distant target cell, and this includes the secretion of the cytokine into the bloodstream and it traveling through the circulation and then affecting the target cell. And this includes interleukin 6, TNF alpha, IL-1 beta, which are secreted and then travel to the hypothalamus to induce fever. Now these cytokines are involved in inflammation, so we can think of how they have multiple functions in inflammation, and they are going to affect distant target cells rather than cells that are closer to them. Now we're going to look more closely at the role of interleukin 2 and IL-2 receptors in T cell proliferation. So as we can see here, we have a resting or a naive T cell. And if we zoom into the structure of its IL-2 receptor, we can see that it's only composed of two chains, which is the beta and gamma chains. Upon its activation by the antigen and its co-stimulatory signal, which is basically our TCR-MHC binding and B7-CD28 binding, we're going to have secretion of IL-2. And we said IL-2 is going to act on the same receptor. So after secretion of IL-2, we're going to see upregulation of the alpha subunit as well. So now rather than it just being composed of a beta and gamma chain, it is composed of an alpha, beta, and gamma chain, which is going to increase the affinity of the IL-2 receptor to interleukin 2. And this is going to induce furthermore T cell proliferation. Alright, so this figure is sort of giving us the bigger picture of what is happening. Up until here, it is what we have discussed, which is the activation of the T cells through the binding of MHC and TCR and its co-stimulatory signal, which is B7 and CD28, and then proliferation and differentiation. So now that we have our differentiated T cells, they're going to enter the circulation and they're going to migrate into the site of the antigen. Now once they migrate into the site of the antigen, they are going to start performing their effector functions. As we can see here, we have release of cytokines, which is mainly by the CD4 positive cells, and this is going to lead to inflammation, phagocytosis, and killing of microbes. Whereas our CD8 positive cells, which are our cytotoxic lymphocytes, they're going to directly kill the infected cells. And over here we can see that some of the activated CD4 cells actually remain in the lymph node, and they're going to enter the lymphoid follicles and they're going to help B cells with the production of antibodies. Now, the migration through vessels in peripheral tissues is going to help them bind to the endothelial cells that have been activated into cytokines, and this is going to allow the lymphocytes to actually enter into the site where the antigen is. Now, CD4 positive T cells recruit and activate phagocytes to destroy microbes. Since they're mainly T helper cells, they're not going to directly kill the microbes or the antigen. Whereas CD8 positive cytotoxic T cells, they're going to kill the infected cells directly. And we're going to see how they do that in a moment. So lastly, we're going to talk about CD4 positive cells and CD8 positive cells. And since we were already talking about how CD8 positive cells are going to directly kill the antigen, we'll start off with CD8 positive cells. So as we said earlier, CD8 is going to be activated by MHC1. As we said, 1 times 8 is equal to 8. And they are going to induce apoptosis of virus-infected or malignant cells or cancerous cells. And how they do so is through the actions of granzymes and perforins, specifically granzyme B. So what happens is upon the activation of the CD8 positive T cell, if it is a naive one, it is going to require both the MHC binding to TCR as well as the co-stimulatory signal, which is B7 and CD28. However, if it has already been activated prior to that, It only requires recognition of the antigen peptide on the MHC1 protein surface. So after we have these signals, it's going to trigger the release of granzymes and perforins. So what perforins do, they're going to perforate the wall of the infected cell. So it's going to create these tiny pores, which are going to, and then we're going to have granzyme B specifically entering through those pores, and this is going to induce apoptosis of the virus-infected or malignant cells. Now, the clinical relevance of CD8 positive cells is that they are involved in organ rejection as they stimulate the apoptosis of the graft donor cells. Now, as for CD4 positive cells, again going back to the rule, 4 times 2 is also equals to 8, so they're activated by MHC class 2. So we have TH1, T helper 1, TH2, T helper 2, TH17, and that's T helper 17. We'll start with TH17 since it's the simplest. It is going to release interleukin 17, which is a pro-inflammatory cytokine. Now we move on to T helper 1, which is responsible for immune reactions against intracellular pathogens, such as viruses and intracellular bacteria. So these are going to release cytokines that are going to stimulate macrophages, CD8 positive CTLs, and NK cells. And they're going to secrete interferon gamma, which is going to activate macrophages against intracellular pathogens, such as in the case of tuberculosis. And I inserted this from an earlier lecture just to show you that initially this is one of the mechanisms of macrophage responses. So they're going to secrete IL-12, which activates lymphocytes, which in this case would be T helper 1 cells. And these are going to secrete interferon gamma, which acts on the macrophages again and allows them to engulf and kill the antigen. Now we move on to T helper 2 cells, which are responsible for immune reactions against extracellular pathogens, such as bacteria, parasites, and allergens. So when we think of parasites, parasitic infections, we should think of eosinophils. And when we think of allergens, we should think of histamine release. And histamine is released by 2 cells, which is mast cells and basophils. So if we combine all of that together, we can say that T helper 2 cells are going to activate eosinophils and mast cells and basophils. So they're going to release cytokines that activate those cells, as well as they are the main activators of B cells.